So I'm going to be talking to you today about RNA modifications on viral RNA genomes. And I don't have the entire syllabus in front of me, so some of this may be a recap to some of you. But I thought it would be important to kind of share my information so that everyone can be on the same page. So hopefully, some of it may be review. I'm not exactly sure. And also, I was actually just visiting Weill Cornell last week, so maybe you saw me there in person, but here I am on Zoom in North Carolina. Okay, so yeah, I'm at Duke University. My contact information is there, and you can go to hornerlab.org to go to my email, or just type in my name onto the internet and you'll find it. So we know that RNA modifications, and you might know this already, really diversify and expand the genetic code. So beyond the traditional 4 bases, A, C, G, U, there are actually a whole lot more modifications that can happen on RNA molecule on these nucleotides that can really, diversify and expand the genetic code beyond these just 4 letters. And so this is really exciting because it means there's a lot of cool regulation that can happen from a biological point of view. These modifications can occur on mRNA molecules, kind of like shown here, but also on tRNA molecules, ribosomal RNA molecules, and other types of RNA molecules that exist. I'll be talking about mRNA molecules. And we know that RNA modifications are important because, for example, and you're not going to be able to read this table here, and I've tried to put the references below, so I can send these slides and you can have access to them later to kind of look up things. But we know that RNA modification enzymes are dysregulated in many different types of cancers, things like acute myeloid leukemia, hepatocellular, glioblastoma, etc. And so this tells us these are biologically important things that are happening in the cell. And this kind of can make sense because RNA modifications can regulate gene expression. And if you think of cancer as a disease of dysregulated gene expression, it makes sense that if you change this regulation, you could ultimately impact cancer. RNA modifications are also known to facilitate immune evasion. And so this is a slide or a schematic from the American Society of Virology that really demonstrates this nicely. And this is one of their teaching slides where they show, for example, a normal mRNA molecule can be inflammatory, but when you put in an RNA modification, in the case of the mRNA vaccines for SARS-CoV-2, this is the N1-methylpseudouridine modification. This limits the inflammatory capacity of this mRNA molecule, and it may also increase its ability to be translated. And so these modifications then are really important for immune evasion, and they allow the development of the technology to make the mRNA vaccines. And so if you don't know this already, this is, of course, what the Nobel Prize was awarded for in 2023, really the idea that these nucleoside-based modifications were what the key to being able to allow us to make kind of these in vitro transcribed RNA molecules that we can put into people when they're encapsulated in lipid nanoparticles and generate then functional immune responses and express their target antigens. And so because of the importance of RNA modifications, there have actually been the importance and also probably the untapped drug marketability and kind of economic space. This slide is a little bit old, but there are a number of different companies that have been developed that target RNA modifications and explore RNA epigenetic concepts. So I'm showing a few of those here. And so, for all the students, I think it's important to realize like learning about RNA modifications can get you a job later. And this is a huge space that have done an enormous amount of fundraising to raise capital to develop drugs that target RNA modification enzymes. Okay, I'm going to be talking about M6A today, and just to make sure we're all on the same page, again, any RNA molecule can have a variety of RNA modifications on it, as I'm showing you here. And M6A is just one modification, and I'm going to base everything today off of M6A, but some of these concepts, of course, could be applied to other RNA modifications. So this modification has a machinery that adds it. So an enzymatic complex, metal 314 in mRNA, and metal 16 also adds M6A in specific RNA molecules. But typically, when we think about M6A and mRNA, we're thinking about metal 314. There are also enzymes that remove the modification. And so this sometimes is thought of to be a dynamic modification that could be, toggled on and off in different contexts. The modification can be read by YTH RNA binding proteins. So for example, YTHDF1, 2, or 3 can actually bind to the modification when it's present in an RNA molecule and then do a function. These proteins can do things like help stimulate translation or promote RNA degradation. And so what this modification ultimately is kind of like a signal for RNA binding proteins to find the modification and then do some function. So whatever RNA binding protein that is, that could have a different function. This modification, of course, could then also repel RNA binding proteins that could have other important functions. So those are the proteins that kind of do the business of regulating M6A at the basic level. And I'm going to be talking today, which I think is relevant to your class, about kind of how to identify and map M6A ultimately on viral RNA molecules. And I wanted to start by kind of giving you a little bit of the landscape of how we can identify and map M6A and the position on the RNA molecules. So the most kind of original method that Chris Mason, Sammy Jaffrey collaborated to identify, along with others in the field, was what we call this MRIP-seq method. This is an antibody-based method to pull down RNA molecules that have the modification. This is kind of, 1.0, let's say. There are a number of other types of modification mapping methods that have come across, that have been developed since these papers came out in 2012. And I think in your class, you're talking about things like nanopore sequencing, and this of course can also be applied. And I'm not going to go over all of these, but I will be talking about a few of them today. I just kind of wanted to tell you that there are kind of updates to the technology over the years. However, there's still some power in this kind of original M6A mapping method. So let me show you what it can do. And so this M6A mapping studies were able to identify a consensus motif in RNA molecules. So when you sequence the bound RNAs and compare them relative to the input and look for changes in the kind of IP over input, you take that sequence, you say, is there anything in common? And you got the consensus motif. We knew what this motif was because that had been determined biochemically. And then we also can make something what we call a metagene plot of M6A distribution over a typical RNA molecule. And it would look something like this, where the M6A peak density appears to be enriched near the end of the gene or near the stop codon on the last exon. And there's also this peak over here, and that actually is due to a different modification called M6AM, which cross-reacts with the M6A antibody. So it has a 2' methylation, so it's a little bit different. And, recent papers have really started to demonstrate what determines which one of these consensus motifs in any given RNA molecule is modified versus not modified. And it's likely that things like the exon junction complex or other RNA binding proteins, if they're bound to RNA as it's being transcribed, that's when M6A is added. It can limit the ability, for example, of M6A to be added to RNA molecules in various places. We know that RNA binding proteins, so Teo was talking to you about VRMA, can actually help maybe promote some kind of methylation or targeting. And there's also some evidence that specific histone modifications could also play a role in, why one of these consensus motifs would get modified or one would not. This sequence is not unique. And so it seems to be that the default would be that any one of these kind of GGACUs would get modified, and only if it's bound by a protein would it not get modified. My guess is it's probably a little more complicated than that, especially under some sort of dynamic settings like a viral infection or immune response, stress response, but this is the best data that we have. We also know that the methylation level of any individual M6A site varies, and I'll show you a slide about that in just a minute. So work in my lab has actually tried to define M6A, done M6A mapping on the host RNA and also on viral RNA. And I just want to show you an example of what that looks like on kind of the cellular RNA. So we did an experiment where we infected cells with viruses, and then we did this MRIP-seq approach where we extracted mRNA molecules, we fragmented them, so that makes them about 100 to 200 nucleotides. We then do the IP with M6A antibody and then do sequencing, and we look for, this kind of peak detection. And within a peak, we're looking for the presence of this motif. You can probably see something like this by your eye, but we actually call it computationally. And it could look something like this. And we found, for example, that it looks like a virus makes a new peak in this one RNA molecule. And so you can see this by your eye, but of course we can call this computationally. There are 4 M6A motifs present there, but we don't know which motif is modified. It's likely that it's more than one. And so this is the kind of a limitation of this technique. Another big, this kind of shows at the bottom. So another big limitation of this technique is that, in fact, if this molecule would change gene expression under a dynamic setting, for example, you'd have more RNA copies because it's transcriptionally activated, you actually have to normalize this kind of like purple bar to this like black input bar. And that normalization hadn't typically been done in the field. And so we actually worked with the Mason lab, or they mostly did it, and we kind of gave some input in how to measure these changes in peaks. So you kind of have to figure out, if you change your gene expression, you have to divide by. And so these methods have been used to map M6A profiles and really define many aspects of the function, but there are some limitations, and I'll kind of show you a little bit about what those are here. And so new methods came out in the last couple of years. I'm only showing one of these new kind of antibody independent M6A mapping methods. So this one is called GLORY, which is glyoxyl and nitrite mediated adenosine deamination. And it takes advantage of the fact that adenosine can be deaminated into inosine, and that looks different when you do sequencing because it's read as a G. And so you can actually do an experiment, what we call a protection. And so I'm not going to get into the chemistry, but there are chemical protection steps that you can do, and they actually will protect your M6A containing mark. And then you can do a deamination. And then, if it's not protected, it gets changed to an I, and then that's different by sequencing. And so this technique has worked really, really well to giving us site-specific resolution of M6A, including knowing that, for example, this A is modified, but this A is not, because when you do sequencing, and this is like a traditional sequencing approach, RNA sequencing, you can see the difference. And so these data showed us that like most M6A modified transcripts actually have multiple M6A sites. I think this wasn't really too much a surprise, but what was crazy is that there are some transcripts that have more than 60 M6A sites on the transcript. So there's a wide variety of how much M6A. There's an average of 10 per genome, but again, the diversity is really interesting. And, let's see, this is 2,000 genes have greater than 20 M6A sites. Some, again, have more than 100. So there's would be highly regulated when you would, for example, change expression of the machinery. We also learned that the distribution of M6A sites within peaks was maybe not what we had hoped. So I had showed you the peak data before, and these experiments showed that in any given M6A peak, there was an average of, let's say, 5 M6A sites. So that kind of supported some thoughts that happened in the field. But I think this graph over here, to me, was really interesting, because what it showed is that actually 45% of M6A sites are within a peak. That's great, but over 50% of sites were not within a peak. And so that tells you that those peak-based calling algorithms actually weren't really good enough to really tell you, only worked 50% of the time. In addition, these showed us that within a peaks, 30% of peaks had 0 M6A sites. And so that really tells us that these kind of antibody-based mapping methods aren't good enough to give us really high resolution data. And sometimes they're actually completely wrong. They also told us, this paper also told us, that the modification ratio of sites within a peak can vary. So if you have a peak that looks like this, and it actually found that within this peak, there were 3 different M6A sites. Sometimes they were modified at 38%, but sometimes 68%. And so likely that's not because it's on an individual, on our RNA molecule, that's all different, but this is actually a combination or a snapshot of all the RNA, excuse me, of all the RNA molecules for this particular transcript or gene. So let me show you what that looks like. And this underlies some of the challenges to mapping RNA modifications. So if you have, I've just kind of drawn a generic RNA here. It may probably have a cap and a poly A tail. Maybe it had been spliced. If you profile the modification sites, it might look like this, but we don't actually know from antibody-based methods if M6A marks every RNA copy. You know, there aren't going to be 3, but imagine that this is a group of them. We don't know if M6A is heterogeneous across an RNA pool. So does it look like this, or does it look like this? And because we can't actually distinguish between these two methods, we don't know if M6A could tag an RNA molecule for a specific function. For example, maybe this molecule on the top would do something interesting that would be different than this RNA molecule on the bottom. And we also know that M6A antibodies don't always interact well with heavily structured RNA. And this is actually important for viruses. This here shows a shape map structure of part of the hepatitis C virus RNA molecule. You can see it's heavily structured. And so if these antibodies have challenges in recognizing structure or differential preferences, then that will be a problem in using the antibody. And so that gets us to kind of like the meat of today's lecture that I'm going to be giving you. And it's really about the challenges to mapping and defining M6A function in viral RNAs and how best to construct M6A maps in viral RNA molecules. Myself and a postdoc in my lab, Matthew Thompson, wrote a perspective piece that just came out in the recent issue of RNA. And so if you want to read what we wrote there, but I'll kind of give you the snapshot here. And so since this is not a virology course, but maybe you heard a little bit about viruses, I just want to mention that viruses will all have RNA at some point in their life cycle, but the genetic information that is kind of the transmissible part of the virus could be different. So there are viruses that have DNA genomes. These genomes often are processed very similar to cellular, to DNA, but ultimately they have to make an mRNA molecule. Retroviruses can start with a positive strand RNA molecule, and this gets reverse transcribed and can become integrated, and then that will make an mRNA molecule. And then there are a number of RNA viruses, either positive strand RNA viruses, so that means their genome is a positive sense polarity, negative strand RNA viruses, or double-stranded RNA viruses that have genetic information, and then this genetic information has to get converted into an mRNA molecule, which makes a protein. And so these viruses all differentially encode genetic information. And if it's a DNA virus, how it works is probably typical to a typical cellular mRNA molecule and how it's made. There are some exceptions. RNA viruses will have to be different because there's no DNA. And so I want to give you just a brief primer of the genetic diversity that can exist between positive strand and negative strand RNA virus genomes. Because if you were going to, for example, map modifications on these viral genomes, you have to know that there are actually differences in the sequence. If you go to GenBank or somewhere and download the sequence, you actually have to know this information as well. So a positive strand RNA virus genome, when it enters a cell, the genome is the mRNA, but it also has to get replicated. And so this molecule makes the RNA-dependent RNA polymerase. It makes a negative strand copy, which is a template, or this is the template for anti-genome synthesis. And then this can be converted into mRNA, but also this is mRNA. These two molecules at the top are the same. And this is a molecule that gets packaged into virions to make progeny virions. So this is what it looks like for some positive strand RNA viruses. For others, they also encode what we call subgenomic mRNAs. So these are shorter molecules that are made from the negative strand copy. And these subgenomic RNAs can also make proteins. SARS coronavirus 2 has subgenomic mRNA molecules. Flaviviruses do not, hepatitis C virus does not. So just getting this sequence isn't enough to know, which one of these it happens. You have to know a little bit about the virus. Negative strand RNA virus genomes can be what we call segmented or non-segmented. So within a virion, there could be one molecule or there could be, 10 different molecules. So flu is an example of a virus that has 10 different RNA molecules, where other viruses will just have one RNA molecule. And for these viruses, the thing that's in the virion is the negative strand genome, kind of in this you can't tell, olive green color. And this has to get copied, when it can get copied into the full-length positive strand complement and then copied back. But to make protein, it has to make mRNA. And so the mRNA has to be kind of capped and polyadenylated. And viruses have unique strategies for doing this, but this capping and polyadenylation, makes this molecule then different from the kind of what we call the genome copy versus the mRNA copy. And so these in theory, these segmented can be present in different abundances. And influenza, that allows for what we call reassortment and for kind of new flu epidemics and pandemics that occur. But it also means, that each one of these segments could come from somewhere else. It can be mixed. It can have different mutational profiles. And negative strand RNA virus molecules can also exist in non-stoichiometric amounts. And so this is the genome of a paramyxovirus virus, kind of shown at the top. And you can see that you actually have like more copies of the N molecule than this L molecule. And you don't have to know what these are to know that if you have different numbers and you're doing a sequencing experiment, you're likely to get more N than L. And it may have nothing to do with, how much modification profile there is on that particular RNA molecule. Viral RNA molecules don't always look like cellular RNA molecules, and that can make doing things like a nanopore sequencing run actually challenging. So for hepatitis C virus, this virus has one long RNA molecule. I'm kind of have a schematic of it shown here. It's about 10,000 nucleotides long. It doesn't have any poly A tail, and it doesn't have a traditional M7G cap. It has a metabolite cap in some cases. And this is a virus that doesn't go into the nucleus, so it doesn't use the normal cellular machinery to do transcription, but instead uses this viral protein to copy the viral template. So that's the RNA-dependent RNA polymerase. And so this is just one example of how viral RNA molecules can look different than cellular RNA molecules. And so as you think about sequencing them, you want to know that they look different. RNA virus populations also encode a great deal of genetic diversity. My guess is you probably don't need to know this, and you already know this because we've all seen in SARS-CoV-2 the kind of different variants that have come through over time. And this is really because RNA viruses replicate as a quasi-species, so they all look a little bit different, even different than your kind of parental sequence. This genetic diversity probably contributes, there are actually experiments that show that it can contribute, for example, to pathogenesis. Obviously, it would also contribute to immune evasion. And so one other important feature about viral RNA genomes is that one molecule could, in theory, have different roles in the viral life cycle. And so this may be different than a typical RNA molecule whose job is just to make a protein, let's say, or encode that information. In viruses, an RNA molecule can be present, let's say, in the virion. It could be the template for translation of the proteins. It can be involved in replication, and it can be the RNA molecule that gets packaged. And so that means, you have 4 different potential RNA molecules, maybe 4 different modification profiles. And so I want to mention that, sometimes it may seem that your viral life cycle maybe appear incompatible with how an RNA modification could be added onto it. So for example, you might know that canonical methylation takes place in the nucleus co-transcriptionally. Hepatitis C virus, for example, replication is in the cytoplasm. It doesn't use RNA polymerase II. I've already mentioned it uses this RNA-dependent RNA polymerase. So how could that work? And it's really because viruses can relocalize M6A machinery to target RNA and to target their RNA. And so if we look at, a broad overview of the M6A methyltransferase complex, Teo was talking to you about VERMA a little bit. Metal 3 is a traditional kind of enzymatic subunit, but it's actually part of a larger complex. And these molecules all potentially have roles in targeting. And so viruses can, for example, any viral protein may be able to bind to any one of these things as it's being actually, for example, translated in the cytoplasm, and then target this to the viral RNA molecule. So viruses are really good at changing how things work. So that's important to keep in mind. Okay, so what do we want for M6A viral RNA maps? Ideally, we want full-length viral RNA molecules. So if 10 kilobases for hepatitis C virus, 30 kilobases for coronaviruses. We'd want isoform-specific information. So if you have a subgenomic RNA molecule versus a full genome RNA molecule. We'd want single nucleotide resolution. Ideally, you'd want it specific to viral life cycle stage. So the incoming RNA, the translating RNA, the packaging RNA, the replicating RNA. And you want to do all of this in virus-relevant cell types, ideally maybe from like a person. And so this is actually a pretty big ask. And so we're not here yet, but I'm going to tell you, just in the last few slides, kind of how we're thinking about how this could be done. Okay, so if we want to map M6A sites on viral genomes, we really want to think about for these kind of four main concepts. What are the profiles of M6A? So can we get this long enough read length to get the information we want with enough resolution so we could know, whether it's two M6A sites on one molecule or one on different molecules. I'm only showing M6A here, but presumably there are other modifications. Ideally, I'd like to know what the whole profile looks like, right? Maybe nanopores will be able to do that one day. And people are definitely working on that. We'd want to know which A is methylated in any particular sequence. And so that really requires, again, this resolution, and then it requires better methods than antibodies. We also want to know the abundance. So is it 40% modified? Is it 100% modified? What does that mean? And we'd also like to ideally isolate, different viral life cycle stages. And that, and maybe even be able to see them in the cell. That's a pretty big ask. So those are the things that we're thinking about in my lab. And I'm going to show you a little example of how we kind of have done this. And I'll start for some of our original studies, which, aren't next generation. They're kind of what we did first and how we're kind of thinking about going forward in probably just the last 5 minutes. So we have done things like use antibody-based methods, which we know are imperfect, but combine them with maps of where RNA binding proteins that sense M6A bind to RNA. So if you have a MERIP peak and it's also bound by an M6A binding protein, that's a high confidence site. And so doing that, we've in red shows a kind of a MERIP profile across the hepatitis C virus RNA genome. But if you overlap that with parklet data, and we did this for all 3 reader proteins, but if you, and so this is for 2 different reader proteins with an M6A set. We have these 2 really high confidence sites, and we were able to make mutations in this region here and activate M6A and figure out that it helped regulate viral RNA packaging. So a combination of M6A mapping approaches here was really essential to identify which one of these sites to mutate. These other sites could be noise. We don't know that yet. Importantly, in these kinds of experiments, we want to do controls. So if you can knock down the M6A machinery and you do a MERIP now coupled with qPCR and you lose your relative M6A on the virus, just like you do on a cellular RNA, that means it's probably real. And so those are things that we like to think about when we're doing mapping experiments. So how can we map M6A and viral RNA? I told you we can combine those kind of methods. And if we think going forward, there are 3. One is using a technique that we call molecular recording. The next, and I'll show you what that is. The next would be using these kind of M6A protection-based sequencing strategies like GLORY. That's one of the methods. And the last would be doing direct M6A calling via long-read RNA sequencing, which is kind of what you're talking about in the class. So molecular recording actually has some ability to give you single RNA molecule information. And so this technique is pretty cool. It uses the YTH domains, and this was developed by a number of groups and applied most recently by my colleague Kate Meyer here at Duke, where you can fuse something that binds to M6A to an RNA editing enzyme, like an ApoBec or an ADAR. And then when you do RNA-seq, along with a control that doesn't have this fusion, you can then directly see where these things are binding. And this is from cDNA sequencing in the end. And so this is, cost is really pretty easy to do. You don't need a lot of input material. And it only works as good, though, of course, as these YTH binding modules are for sensing your modification. But, this figure from this paper, it looks pretty good, where if we kind of look at known M6A sites and we kind of overlap them, there's pretty good overlap. This, hopefully you can see this part. It's really small on my screen, but ultimately it does allow you to potentially give you single molecule, especially if you couple it with long read sequencing, like a PacBio, or even Nanopore, just cDNA sequencing. It may be able to give you relative % modification if you sequence deep enough. And there's a potential for a kit or in vitro applications, because if you could purify this enzyme in vitro, you can just add it to your RNA that you isolate and then in a test tube. You don't have to do it from cells. So that has a huge advantage because you could do it now from a patient sample. So I really like thinking about how we could apply this approach to study M6A and viruses. I've already mentioned that GLORY is a great way to do absolute quantification of single base M6A methylation, and it can really identify which A is modified. And so I've showed you these slides before. Some advantages are that it really gives you this residue resolution. It gives you absolute quantification. It requires a low input, so you can do it on precious samples, let's say, but it does require high read depth, and that can be a cost limitation. And it's not, while it's not impossible, there are some specialized reagents and protocols that, you know, might be difficult for some people, including the fact that many of these chemicals can sometimes totally shred your RNA. So it can be hard to actually sequence the molecules that come out at the end and get meaningful information. And then the kind of second to last slide I have here is that a number of groups that we've collaborated here with Matt Weitzman at Penn have combined things like a MRIP with a nanopore-based approach to map M6A. This is on adenovirus RNA molecules. So adenovirus is a DNA virus. But with this, we had to do things. We had a parental cell and a metal 3 knockout cell that we infected. We did a MRIP-seq to identify where the peaks were, and then did a direct RNA-seq to get the different RNA isoforms. And when you compared your wild type to the metal 3 knockout, you could look for current changes that would be predicted M6A. This is a virus that's highly spliced. You really can't do an M6A map with regular kind of MRIP-seq approach. You need this information. Nanopore will require you, but in some cases, to have a read length and a depth of sequencing long enough to get the profile. So if your RNA molecule is 10 kb, how do you get a direct RNA sequencing of 10 kb at a high enough confidence? The typical read profile is a little bit shorter than that, although you can get it. If your virus does not have a poly A tail, you got to do something special to get it to stick to the nanopore and be read. It is a prediction-based method, and you need controls, for example, an in vitro transcribed RNA, which Teo is talking to you about. Okay, so I will end then by just giving you a summary. So I've told you that RNA modifications can really provide a layer of regulation to the genetic code. Many methods have been developed. There are limitations. Viral RNA molecules are unique from cellular RNA molecules, and that makes sometimes mapping M6A in them a little bit more challenging. There are a number of few things that we're thinking about when you really want to map M6A in viruses. And ultimately, probably combining and integrating data from multiple methods is what's really going to be needed to define M6A function in a viral RNA molecule. So I actually don't think just one method will be enough. So I will end there, and I'm happy to take any questions.
Great, we'll do some clapping. Thank you. All of us here. Great. Thanks. Good to see you again. Hopefully a lot of you caught Dr. Horner's talk last week, which was elaboration on a lot of the viral components. Any questions before we do last part of the class for today?